Well, welcome everybody to Bodega Marine Laboratory and also to the Coastal and Marine Sciences Institute at UC Davis. I'm John Lodge. I'm Director of BML, Bodega Marine Lab, and also Professor of Oceanography. Pictures here. Established in the 1960s, Bodega Marine Laboratory has developed a substantial research community, including almost 100 staff and students and faculty who are based here, resident in Bodega Bay, in addition to a bunch of colleagues who are resident in Davis, forming quite a strong research community. This notable research and education, it happens here, is founded on our long tenure, but also our critical facilities. Probably first and foremost is the seawater system that brings the ocean into this building through multiple research aquaria for conducting research. But in addition, the facilities that take us out into the ocean, onto the shoreline and offshore. And thirdly, the long-term tenure of tracking this place for several decades now in a time of climate change. The impact of BML extends well beyond Sonoma County and is greatly enhanced by collaborators and visiting researchers, none more valuable than long-term colleagues who have worked here also for decades and keep coming back, who studied organisms in this place over a long period of time, also using the facilities and grounded in the scholarly community, developing new insights and publishing groundbreaking papers. A great example, of course, is Don Michaels, Dr. Don Michaels, who has been associated with BML for 50 years. After founding the Crab Lab, who thought of that name, at Colorado State University, he's continued to intertwine his career in the studies of invasive green crab, expertise in integrative biology with BML and with this collaborative community of colleagues here. It's special that Don is here today, as well as his wife, Vicky. So, it gives me great pleasure to welcome you to the annual community lecture in the Dr. Donald L. Michaels Distinguished Lecture Series. While it's just the second year, it already feels like one of the rites of summer, a celebration of science and exposition of the impact of research in this changing world. The Michaels Distinguished Lecture brings us together and welcomes into our community a leading researcher who integrates physiological processes up through and across many levels of biological organization and disciplines to address grand challenges in marine and coastal sciences. Ben encourages us, to lift our heads and look up for a while from our individual and even our collaborative research to explore new and bigger ideas. The joy in this annual rite is enhanced by the role in the post-COVID revival of BML. It was really quiet here a few years ago. Although conceived and partially planned prior to COVID, these lectures and these multiple interactive meetings that have been going on for a few days could only be implemented post-COVID. And as such, it reminds us of the immense joy and the pleasure and the importance of spending time together. The Dr. Donald L. Michaels Distinguished Lecture Series was established through generous gift from Vicky and Christopher Michaels to benefit BML and the community surrounding us, as well as to honor Don's scientific career. And they welcome you to join them in continuing to support this lecture series going forward. Now it's my pleasure to introduce Dr. Adrian Correa from the University of California, Berkeley. Dr. Correa is an associate professor in the Department of Environmental Science, Policy and Management, and co-director of the Berkeley Center for Ocean Futures. He's also associate director of the Gump Station in Moorea, French Polynesia. Talk about two different interesting places. She received an MS in Conservation Biology and a PhD in Ecology and Evolution from Columbia. She was a postdoc at FIU and OSU, you know the acronyms, right? Before becoming assistant professor at Rice University in Houston, Texas. Last year, she joined UC Berkeley. We're excited to have her as a neighbor and a future marine colleague. Well, that's our hope. Dr. Correa and her group research how microorganisms influence the animal and plant hosts and also ecosystem processes under environmental stress. They're focused on the factors that influence disease spread in and through communities and how species interactions can influence coral symbioses. Her work spans from microbes to ecosystems and incorporates omics tools, microscopy, and culture-based approaches to understand microbial contributions to ecosystems. Much of her work is in coral reef ecosystems and the diverse microbial assemblages that corals host. The unique aspect of Dr. Correa's work is the virome. I hadn't heard that word before, but guess what it is? The assemblage of viruses that are associated with hosts like corals and how that influences coral health, including coral bleaching. She's a leader in understanding coral disease epidemics, transmission of microbes through consumer vectors, and virome on coral and the virome on coral reefs. It's an honor to welcome you, Adrian. And I think I speak for all of us in saying that very much looking forward to your lecture. Briefly, I'll tell everybody to please enjoy the lecture and make sure to stay afterwards for the reception. So I'm talking to you. Everybody is welcome, no matter what your link to us is. Come and hang out for a while and talk a bit afterwards. Adrian, up to you.
Is this working? Can you all hear me? All right, wonderful. If you can't, please start sending me a sign. I will try to speak up. Thank you to everyone. Thank you for having me here. Thank you for the wonderful introduction. Thank you to the folks at BML and in particular, Dr. Brown, Ellie Fairbairn, John, many people, the entire BML community for really being wonderfully welcoming and engaging over the past few days that I've been here. It's my first time out in the Bodega Bay area. Wow, is it wonderful here? I want to come back. I want to come back to BML. So, in particular, also thank you to Dr. Don Michaels and his family for, Don, for your work and your participation here at BML for so long. And for starting and participating in this amazing lectureship series. I think it's wonderful. It's been an amazing opportunity for myself and my lab group to be able to come out and meet everyone, especially since now we are neighbors and we can come and do research here. And I really hope that we will do that quickly. And form collaborations. And also, I'm so excited that everyone here in the audience today, particularly, community members of the audience that came out, thank you for coming. Thank you for caring about BML. Thank you for being interested in science as a way of knowing about the world around us. Thank you for being open to checking out some research tonight, some interesting things about coral reefs. So I hope that we get to chat after this is over. And yeah, I'm just excited to know you all. So I am excited to talk with you about just basically a splash of some of the work that we have been doing on coral reefs in my lab group. And hopefully many of the components of this image will become clear to you as I'm talking. Maybe it's gonna move forward. Nope, oh no, what have I done? Well, it's gonna be a short lecture, folks, or I'm gonna freewheel it. Thank you, thanks, Simon. Okay, so coral reefs are amazing. Has anybody been on a coral reef? You can raise your hand. Okay, yeah, or if you've watched a documentary about a coral reef, hopefully you are in love with them the way that I am, and maybe you'll be even more in love in the next hour. They harbor so many species. 25% of known marine species live on or use coral reefs. They are really important economically. We get pharmaceuticals from coral reefs. They support vibrant fisheries. They support tourism. They support even, they form the land that some people in the world live on. So, and they also help protect beaches from erosion, from wave action. There's so many things that coral reefs are doing for us that we often don't pay attention to at all. This adds up to $2.7 trillion US dollars per year in terms of goods and services. So it does not matter who you are or where you live, you need coral reefs. They're amazing. And you can see some right here. Hopefully I didn't just mess up the PowerPoint again. Unfortunately, coral reefs are in a very difficult time right now. A lot of the corals that make up the reef, and I'll talk with you about that in a moment, are dying, and they're dying because of human activities. And one of the main activities that is really impacting corals on reefs is warming seas due to climate change. So you may have heard about bleaching, which, this is a timeline of some different coral species, and higher up on this graph means more corals out on the reef. And as you can see over this long time scale, the amount of these corals out on reefs has been going down. And there are some major things like coral disease and coral bleaching that go along with human activities and human impacts that are driving down the amount of corals that are on reefs, and that drives down the amount of benefits and services that reefs can provide. Okay, so here is an example of what it looks like. This is a picture of a coral reef before bleaching, and then the same reef when it was in this bleaching state. So this is one of those states where when seas get too hot for too long, we see this effect, and this is corals being sick. They're still alive, but they're sick. And so unfortunately, because of coral bleaching and other things that are happening, sometimes overfishing or overharvesting, pollution, we're seeing a lot of reefs decline, and we're very concerned because as these events that cause coral death continue and they intensify and become more frequent, we're worried that we're just not giving corals a chance to bounce back, and that there will just be too many of these in a row, and over, kind of very near-term timescales, we could lose a lot of coral reefs in the world, or at least they won't look like what we are used to thinking of when we watch a documentary or when we swim on a reef. There can still be some coral colonies out there without it being a reef in the way that we think of coral reefs today, right? So scientists are concerned about this, managers are concerned about this, some politicians are concerned about this. I hope you're concerned about this. This is something I care very deeply about and want to do something about. So let's jump back for a second. I'm going to talk with you a little bit about what reefs are and how they work, so that we can talk about maybe one thing that we're thinking about in my group for how we might be able to help. So a coral reef, when you imagine a coral reef, you might imagine a bunch of different things. One of the things you might imagine is this huge structure that can be hundreds of kilometers long that you can see from space, like the Great Barrier Reef. You can see it from space. It's the biggest thing on Earth made by living organisms that we have. So here's a teeny part of the Great Barrier Reef surrounding this island called Heron Island, which is a really amazing place to work off of Australia. And so inside, this whole inside part is reef flat, and then there's a reef crest, and then there's more of the reef out here that you can't see. So there's this giantness to reefs. But when you start zooming in to a little patch of this reef, oh no, I hope I didn't just do it again. Oh, yes, okay. Then you can see these individual coral colonies that make up that giant reef framework, or that reef expanse that I showed you. And there are other organisms that are important on reefs and that are there helping glue reefs together. But on a healthy reef, you've got a ton of these living corals, and that's what's building this giant structure. And if we zoom in even more, maybe onto one of these branches on this coral, what you will see, this is the fundamental living thing in a coral. That's a coral polyp. It looks kind of like a hand. The mouth is in the middle. And each one of these little hands, as a coral gets bigger and bigger, there will be more and more of these polyps, and they all live together. They kind of share certain things within the coral. But the reason that, so this is all like the same individual, all these different little polyps. And this pigmented color in here is actually because of a microbe. So a microbe is something like bacteria or viruses or other kinds of things that are so small that you can't see them with your naked eye. You may have heard a lot about microbes in terms of pathogens or getting sick, like COVID-19 is a virus, or you might have heard a lot about the microbes that live on and inside your own body, like the human microbiome, does important things for our digestion and other parts of our health. So microbes live in and on all animals and plants, and they do important things that make animals and plants the way that they are. So, if there's an animal or a plant, there's a bunch of microbes that live on and in it. And so one of the microbes that's super important for corals is a dinoflagellate. It's a eukaryote. So it's in this family called Symbiodiniaceae. And I promise that is, I hope, the craziest word that I say to you tonight. I'm going to say it a lot, Symbiodiniaceae. But it's just this kind of microbe that gives coral polyps all this color. So there can be, they're nice microbes. There can be 1 to 2 million of these microbes per square centimeter, so tiny, per square centimeter of coral tissue. So that's how they give corals the color, is that there are just so many of them inside of coral tissues. So when you're looking at a nice, bright, beautiful coral colony, you are seeing many, 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 many microbes. And what they are doing is they are photosynthetic organisms, so they take in sun's rays and they make sugar, and they actually are very generous with their sugar. Often they dump out almost all of it to the coral animal tissue, because coral's an animal. So they dump out a ton of that sugar into the coral animal, and the coral animal uses that to build those really big reef frameworks. That's okay. And then in return, because this is a partnership, and usually it's a good mutualistic partnership, so they both benefit, the coral is releasing its waste products, which the Symbiodiniaceae really need as nutrients to grow, because it's almost like a desert in these ocean waters for these corals. So when the corals give these nutrients, the Symbiodiniaceae are really happy. It's like getting fertilizer. And the coral also gives this stable place for the Symbiodiniaceae to live and grow on the reef, protected. So one thing I want to make sure you know is that, okay, there are a bunch of these Symbiodiniaceae. They look really similar to each other, but when you look at their DNA composition, they can be extremely different. And that matters because when some of these different, strains or groups, when they have DNA differences, and then those different kinds of Symbiodiniaceae are in one piece of coral versus another, it can change a lot of things. It can change how fast the coral grows. It can change whether or not the coral can handle high temperatures or not, or the extent to which it can handle high temperatures. And it can influence health or resistance to certain kinds of diseases. So we care a lot about what kinds of these Symbiodiniaceae are inside of corals, and whether or not corals can change out the dominant kind of Symbiodiniaceae they have in response to a change or a stress, and how that impacts whether or not the coral stays alive. So here's bleaching. This is the same picture of a reef across time. Here's this a whole bunch of different corals growing here. And now you can see a cross section through the coral animal. And you can see most of this is white, because as corals grow, they actually make a rock, which is really cool. They make limestone, and so it's just this outer bit that's the living coral tissue, and the Symbiodiniaceae are in there, and then all this inside is limestone rock, making these three-dimensional frameworks that we love to snorkel on or see. But the thing is, so corals really need to have those Symbiodiniaceae inside their tissues to live in shallow waters, and that's a risk. Most of the time it's great for corals because they can build those big reefs. But sometimes it's risky when the environment starts changing too fast and too quickly. And so when that happens, it can get too hot for this partnership, and then that breaks down in what you may have heard in the news as bleaching. So with bleaching, I just want you to know the coral animal's still alive. You can see the coral polyps here, but it looks white now because it's lost the Symbiodiniaceae, and you can see this white limestone through the living coral tissue. So what can happen when bleaching occurs? The coral is stressed now. It's not getting food. It might be attacked by other things living on the reef. It might not have enough energy to keep on growing. So it needs to get Symbiodiniaceae back to continue to live. If temperatures don't go back to normal and it doesn't get its, well, if temperatures do go back to normal and it gets its Symbiodiniaceae back, the coral can keep living and keep covering a lot of the reef bottom. But if temperatures don't go back to normal, or it gets attacked by something else, or it can't recover its Symbiodiniaceae, then what you can have is a whole bunch of dead corals out on a reef space. So this different color of brown, this darker brown, these corals are dead now, but there's still the limestone structure. The reason it looks brown is because algae have come and grown on top of that limestone that's left behind after the coral animal has died and gone away. And what is so challenging is it's difficult for baby corals to come and start growing here. So it's difficult to go back from this. What you can have is a lot of algae on your reef, but it can be really hard to go back from that algae-dominated state to being a coral-dominated reef. And then if it's, algae has its place on the reef too, but if you've just got mostly dead corals and lots of algae, eventually wave energy is going to break up all these pieces and it's going to get washed away. And then there's not as many homes for the things that need coral reefs to live. Okay. And the other thing I want to tell you, surprise, it's not just the Symbiodiniaceae that are living in the coral and doing, impacting its health and doing things. There's a whole mess of other microbes that are also important and also live in the coral and influence its health and things. So it gets very complicated very fast. And these microbes, all these lines are just trying to show you that not only can each of these kinds of microbes interact with the coral animal, they can interact with each other. Things can get infected by different kinds of viruses. It's a complicated living set of things. So my lab loves to study all kinds of different parts of this in terms of the viruses, how virus infections might infect the functioning, the health, different aspects. So I'm going to show you one piece of this today. Okay, so I've been showing you these ways that corals can be harmed and these ways that corals can get sick and die or get better. We have been wondering, and scientists have been around the world have been putting a lot of effort into this. Are there ways that we can kind of carefully intervene to help, right? Can we use what we're learning? Can we use like the basic science, the basic information that we're learning by studying coral reefs? Can we, twist some of that a little bit and come up with some solutions or some things that we can do to try to maybe make corals more resistant to temperature change or more resistant to a disease or better able to tolerate a change, so how can we help? So one of the ways that people have thought of doing this is to go out into the environment to find microbes or isolate microbes to get them to grow in a lab and then to kind of test them and see, okay, if we add this microbe to the coral, if we add that microbe to the coral, what happens, right? Are there some of these microbes that really help the coral be more thermally tolerant that we can now grow in the lab and maybe mix together and then put out, give some extras to corals to make them better able to survive in the near term to stress? You need to do that really carefully because you don't want to necessarily go taking microbes from one place and bringing them to another place and letting them go. But we could think about, can we at least do this in the lab, right? And then we can think about how might it be appropriate to do it somewhere else or out on the reef. So people are interested in this in terms of preventing bleaching or coral death, making corals grow faster, reducing their stress, or keeping them from getting sick. But there are challenges here. So first of all, just can we even do this process at all? Can we get these microbes to grow? Can we figure out which ones have these good effects? And then let's say that we figured that out and we have something that we want to, for all the coral reefs in the world, can we scale up to do this effectively at a scale that's going to matter for coral reefs in the future, right? So those are both challenges. Okay, some of the basic science that we need to figure out to be able to do this is, where do corals get all this Symbiodiniaceae from anyway? Because people have studied a lot about which Symbiodiniaceae are in the coral and can they change, but they haven't studied as much when a coral is a baby and it settles on the reef for the first time, half of the coral species they don't have any Symbiodiniaceae at all. So where are they getting that from so that every coral on the reef has some? And that will help us figure out if we can if we can isolate some of these things and then maybe use them as a probiotic. So this little cartoon is just showing here are some corals, and here is some algae, and here is some sand on the reef, and here's the water column, and all these little dots are supposed to be microbes, different kind of microbes. And color means, a shade of color means it's one kind of microbe. So different colors are different kinds of microbes. And you can see from this that this kind of coral has one type or a set of shades of one color of microbe. This one has these red tone microbes. And then there's a whole bunch of other kinds of things in the sand and in the water column. So these folks in my lab group, especially led by Karsten Krapstra, pushed a lot of these ideas forward. So what we've seen is this cartoon is basically showing you the situation that we see lots of these Symbiodiniaceae microbes in corals. We see some on macroalgae, but the stuff in the water column and in the sand and on the macroalgae, is it like what's in the corals, or is it not like what's in the corals? Like what's in the corals? No. Yes, they are different colors, right? So that didn't help us too much. We were confused. We were like, okay, finally we can see these Symbiodiniaceae outside of the corals, but they are not really the same thing that's what's in the coral. So what does that mean? And how come there are so few out here of the ones that look like they would go in the coral? What are we missing? So this was very puzzling. Here you go. Oh, well, we started looking around on the reef and thinking, how are these microbes getting from corals to other parts of the reef and then maybe getting into baby corals? And we thought, oh, our friend the parrotfish with its curtain of sand. So the next time you walk on a tropical beach near a reef, you can know where some of that sand is coming from. Thank you, parrotfish. So parrotfish bite a lot of things on the reef, including living corals. And then they swim up in the water, they digest that, they have a special body part that grinds up that limestone rock, and then they swim up in the water column and they defecate, or yes, do various other. So we were thinking, could this be like a nature-based solution to delivering the microbes that corals need all around the reef? Maybe it's already happening on reefs. Okay, so now more cartoons. This is a different way of thinking about these coral-eating fishes, right? Usually, if there's something that's biting the thing you're trying to save, like if there's something biting a coral, then you're like, stop it, get away, don't bite it, I want this coral to live, right? That was at least my knee-jerk response. But we are now thinking in a different way about how consumers, these predators or things that bite corals, how they might actually be influencing the rest of the ecosystem. So in here, a minus means these guys are not helping each other out, right? This is a negative relationship. So corals and algae are on the reef. They're resource species. They're things other species use. They are fighting for space. When they're next to each other, they'll do all sorts of things to get the other one away and try to take the other one's space. And algae also have some Symbiodiniaceae on their surfaces, and they also have bacteria on their surfaces. One thing that's interesting is some of the bacteria that are on macroalgae can make corals sick. So they can fight each other, or they can harm each other that way as well. So when coral-eating fishes take a bite out of corals, they are directly harming the coral a little bit, right? Because they just wounded it and took some of the coral away. But they don't tend to eat the whole thing. So they take some bites without hurting them. And when algae-eating fish take a bite out of algae, they're hurting that algae a little bit as well, right? So that's why there are so many minuses. Then there are some trickier fish where they can't decide what they want to eat. They eat some corals and they eat some algae. They're biting everything, like the parrotfish that I was telling you about. So they are hurting various things on the reef when they bite them. So the whole idea has been kind of like, the enemy of my enemy is my friend, right? With parrotfish. So okay. we'll get to that. With the algae eaters. So these algae eaters are eating up the algae, and so people have thought, oh, these are good for corals because they're taking away the algae that is harming corals. So these algae eaters are really helping the corals be the dominant thing that you see on the reef, right? That has been one of the major things that people thought these algae eaters are good for, and that sometimes has included, or often has included, parrotfish that do this. And when people have thought about butterfly fish or other things that eat corals, they usually have not thought, oh, these coral eaters are helping corals be dominant on the reef. They usually have thought, okay, they have a role, but what they do is they bite corals and they make holes in the coral, or they make the coral weak, and then the coral kind of breaks or falls apart a little bit, or it has a hole in it, and so other species can come and live inside that hole and have a habitat, have a safe place to live. So they're helping other species live on the reef by making spaces or apartment buildings, and they can make sand by breaking up those frameworks. So that's the role that people usually think these coral eaters have. But when they're doing all this, as you saw from the parrotfish video, when they are swimming around the reef after they've bitten all these things and then they defecate, could it be that they are also dispersing microbes from corals and maybe from algae around the reef? And could that be having an effect? So that's when we got into this thank you for biting me thing, because it could be maybe you are helping the rest of the population have access to the microbes that corals need or algae need, or maybe by doing this, you're ultimately making certain kind of microbes available to other corals on the reef that they need to take in to tolerate a changing environment. Or maybe the algae, maybe it's even beneficial for algae to get eaten by the algae eater if when this thing defecates across the reef, it's dropping this basically a pathogen bomb, a disease bomb onto the coral and hurting the coral that way. So there are all sorts of interesting additional layers to these kind of interactions between the consumer fish and the algae and the corals that could be going on. So we got really excited about this, and we're excited about it not just in terms of coral reef ecosystems, but just in general, anytime that you have things that can't move, that grow and make habitat for lots of other species, and they rely on a microbe and they get eaten by things that move around and defecate, you could have a similar thing happening. So this could be like oysters, it could be like sponges, maybe marsh grasses, tree roots. Lots of things have these kind of microbial situations going on. So we're generally interested in how this pattern might influence ecosystems and health. Okay, so there's a lot of nice ideas, but first we needed to see, well, do these microbes, when they pass through a fish gut, do they even survive that? Or do they get just crushed up and dissolved away and they aren't alive in the feces? And then, assuming that they are alive in the feces, will other corals out on the reef actually acquire them and incorporate them into their bodies and use them, or not really? So these are the things that we're working on in the lab as a next step. And so just to show you how we got to some of this, like how did we actually test this? What we did was we took a bunch of species we took 4 species that only eat coral. And these numbers here show how many individual fishes we sampled the feces from. So this was a fun project. And then we took 3 species of these fish that eat both corals and algae, and 2 species that eat only algae. And then we wanted to compare it to, well, wait a minute, to really know what feces contain microbes that are relevant, we better get some of the microbes from corals too, so we have something to compare it to. And then we said, okay, well, let's look at the sand and the seawater as well and see what microbes we see there. So it was a lot of samples in French Polynesia. And how do we analyze this kind of thing? You can do it a bunch of different ways. So every time we sampled either a coral or sand or seawater or feces from one of these fish, we would try to look at it in different ways. We would count how many of those Symbiodiniaceae cells we could see in a microscope. And there's this special stain that you can apply that it makes cells that are probably dead blue, and it leaves the cells that were alive not blue. And so when you put it under a microscope, you can not only count how many cells there are, but you can say how many of these are alive in the feces versus dead, which is helpful to know. And then another way to see, okay, are these Symbiodiniaceae cells that come out of a fish still alive? Are they going to survive out on the reef after they make it out of the feces? Is to just try to keep them growing. So we would do a series of filtering steps where we would get the feces, we would get the Symbiodiniaceae cells out of the feces, yay! And then we would put it into this special growing media in these containers and just keep it going for months. And if it kept, if we could still see cells after months, then what we put in that media to begin with out of the feces, it must have been alive and it must have been okay to keep growing. So that's another way to know, yeah, these things are okay after being eaten by a fish. And then we also, because I told you that these Symbiodiniaceae cells, they look similar, but they can be really different with their DNA. We also did DNA analyses to see how different they were and what they were. So for DNA analyses, this is a piece of coral in a tube, in a preservative, so it's got Symbiodiniaceae cells, it's got viruses, it's got bacteria, it's got the coral animal tissue, it's all in there jumbled together. And then you can do some lab procedures where you separate out all the DNA from all these different things. And then you can make a copy of part of the DNA that you want to know about that's specific to Symbiodiniaceae. Only Symbiodiniaceae has it. Or it'll only get that part from Symbiodiniaceae, and then you can copy all the individual parts of the DNA, which is like a fingerprint code that you can compare, and then you can analyze that on a computer. And it basically makes like fingerprints, like a barcode for a product at a supermarket where you can tell like which ones are the same and which ones are different, and then you can compare what each sample has. So that's the scientific gobbledygook of that if you're interested. Of which gene it was. Okay, so here we go. Now I'm just going to show you what we saw, and we'll go pretty quickly. But basically, here are the results. This is on a log scale, which just means the further you go up this scale, it's not changing one by one. It's like a huge amount. So this is to go up this number of Symbiodiniaceae cells per milliliter of feces or sand or water or whatever. Anything, the further up you go, it's way more, not just a little more. So here's sand and water. You can see dead is the blue and live cells is the black. This is counting those cells on the microscope. These big dots are kind of like the average number for a whole bunch of different samples, so you can just look at that. So it's pretty low, like we expected for sand and water. But boom, when you look at what is in fish feces, there's so many, right? It's like, and even in the algae eaters, which was really interesting, but there's a lot more dead than live. So there are some differences between the coral eaters versus the algae eaters, but overall, we were very impressed by the number of Symbiodiniaceae cells that fish poop has on the reef. And we became very interested that, wow, this could be kind of like a hotspot of beneficial coral microbes that corals need on the reef. Okay, and here is just an example of what we saw when we were culturing. So we did a lot of this, and we could just look under the microscope, and the ones that are spinning around, all of these are alive, and so we could see, yeah, they're definitely living, coming through the fish digestive tract. So that was pretty cool. Okay, and this is just a simplified way of seeing from a DNA perspective what was the same, what Symbiodiniaceae in fish feces was like. Is it like what's in a coral, or is it different? So when you see white, these are the names of different kinds of Symbiodiniaceae. When you see white, it means we didn't see any of that. So for these 3 different coral species, we didn't see any of this Fugacium, and we didn't see any of this Breviolum in these corals that we looked at. When you see dark blue, that means we saw a lot of that kind. So this is a code for Poeciloptera and Porites, 2 very wonderful coral species. Lloyd, we see a lot of this Cladocopium kind of Symbiodiniaceae, right? So I'm about to show you what we saw in the fish. You are looking for if you're gonna see dark blue matching up with what we saw in the corals, right? So, coral-eating fish. These are the different species. We see a lot of the same things that we see in corals. Coral and algae-eating fish. It's getting lighter, and there's getting to be more of things that are not necessarily, you're starting to see some differences here, right? And then algae-eating fish, now really most of what it has is, not the same thing. It's got less of this. So we're seeing some definite patterns as what the fish are eating changes. When they're eating less corals, we're seeing less of the Symbiodiniaceae in the feces of those fish that we see in the corals. And then sediments and water, it's kind of interesting. It's got this same one, but it's also got this up here. So interesting patterns in terms of DNA, right? And some things that are matching in terms of, yes, if there are feces of these fish out on the reef and they fall apart and the Symbiodiniaceae in there are alive, those cells could potentially get to other corals and get taken up, right? Okay, and then there was this whole other thing with bacteria where we saw similar kinds of patterns. I'm not going to show you that, but things are interesting from a bacteria perspective too. We did some math, and we wanted to use a bunch of measurements that we'd taken in the field of what fish bite and how big their feces are and how often they go to the bathroom and all sorts of things to see if we could calculate from these other numbers that I showed you how many cells, how many Symbiodiniaceae cells are getting moved by a species of fish on the reef per day. Like how much are they transporting these Symbiodiniaceae cells around? A lot. They're transporting some of these species are transporting over 100 million Symbiodiniaceae cells over a pretty small area of reef per day. So we got very excited by this. It was amazing to see. And then we started thinking, okay, from all the watching that we did out on reefs of where fish are biting things and where fish are going to the bathroom and those things, how often does fish poop fall on a coral? Do you want to know? 91% of the time it falls out on the part of the reef that has lots of coral out on the reef when we measured it. When a fish would go to the bathroom, it would often fall directly onto a coral colony. So, the feces would sink and it would just be like an amazing amount of Symbiodiniaceae cells getting delivered right onto the coral. And even though sometimes the fish feces didn't fall on the coral, it could be that those feces fall apart and then the Symbiodiniaceae make it into the sand or the water and they could get moved onto the coral anyway. So we are wondering, can this process of fish eating little bits of corals and swimming around the reef and going to the bathroom actually be really important for making it so that beneficial coral microbes are available to corals, especially maybe baby corals or sick corals that need new microbes or corals that have bleached. And, yeah, okay. I've gotten too excited. I've talked for too long about this, so I'm gonna speed up. This happens frequently. So yes, Symbiodiniaceae can survive going through a fish digestive tract. Please tell your friends. And now we're trying to figure out now whether or not corals can take up these Symbiodiniaceae cells from fish species and maybe even do better when they do that. So there are some examples where people, even in 1984, were trying to see whether things from feces, Symbiodiniaceae from feces, could go into hosts. And they saw that for anemones, and they saw that for stony coral larvae, but from a giant clam fecal pellet, because giant clams have Symbiodiniaceae too, and they release them. So Taylor, who's here, and you should ask her about it if you see her outside, is leading a project to try to understand whether or not exposure to fish feces can be a source that baby corals can use to take up Symbiodiniaceae. So we have a very challenging experiment going in the lab where we get these coral babies and we give them different sources of possible symbionts, whether it is from fish feces or the seawater or seawater that we know has no symbionts in it. And we're doing similar kinds of methods to try to test this. And then you might have been thinking, well, wait a second, what about when bleaching is going on? You know, the fish are still out there, hopefully, eating whatever they can eat at that time. What happens during bleaching? So Kara's not here, but she is leading a different experiment where we took corals, we broke them into pieces, and because they have all those different polyps on them, we can have like the same coral in two different containers and then expose it to different things. So we took these adult corals, we bleached them using a chemical that will make all their Symbiodiniaceae go away, and then we exposed them to feces from these fish, or gave them no symbionts at all, or we killed whatever was in the feces on purpose and offered that to see what would happen. These experiments are ongoing, and we are learning a lot from trying to do them. They have been tricky because you have to really carefully control everything to make sure that it only comes from feces or whatever treatment. So I hope to tell you about that in the future. But there's this interesting kind of idea that if bleaching is happening and it's patchy, if it's kind of like a medium or a low-level bleaching, you know, here you can see some of these colonies, some of these corals still have pigments, some of them are really bleached. So if fish are feeding, there have been some observations on reefs that some butterfly fish or coral-eating fish, they prefer to bite in a situation like this. They may prefer to bite the corals that are not bleached. And if that's happening, that's really interesting because maybe in these corals that are not bleached under these conditions, maybe those Symbiodiniaceae are doing better under those stressful conditions. And then if they are the ones that get spread around the reef, maybe this sort of interaction becomes really important during stress, more important than during regular times. And you know, you can kind of even think of this as humans use fecal transplants in medicine. You know, maybe the same pattern is happening on the reef. And so, you know, yeah, we're kind of comparing what happens during normal temperature times versus heat wave temperature times to see whether it's an even more important source in these fish species. And so, you know, there are different ways that we could think about, okay, what should we do as we continue to learn more? You know, how could we apply this? We could think about, instead of just doing a coral nursery and coral restoration, maybe there are some reefs where coral-eating fishes have been lost, and we could carefully, with careful timing, you know, grow some of these fish as well and reintroduce them back onto the reef during coral nursery and restoration activities. Maybe that could help. Or you can, you know, getting really wild just to, you know, maybe we need robots that like, we could, even in situations where we can't use a fish, et cetera, you could think about a robot or a drone on a reef that is, if we've made some amazing probiotic that we want to disperse, we could do something like that. Okay, so coral reefs are valuable, but they are threatened. And, you know, we need to think about interventions that we can do beyond, you know, using less, emitting less, et cetera. We can think about some interventions that we can use involving microbes and consumers that might help. Most of the time, we've thought about algae-eating fishes as what's keeping corals dominant on reefs, but coral-eating fishes might also play a role, and there are important management implications to it. So with that, I want to especially thank the team. You know, I have not done this by myself, and I never could do this by myself. It's the team that makes it go, and collaborators and funding. So that's all, and thanks for being here.
Wow, great talk, Adrian. Thank you so much. And thanks for leaving some time so these wonderful people can ask some questions or comment or whatever they would like to do, participate, right? So if you're in the room, just raise your hand. If you're on Zoom, we'll track you on Zoom and take your question, first looking at the room for curious questions.
And if you're in the room, you get to use this really cool box.
We could start in front here. Our friend. That's how it works. And when you speak into, just hold it close to your face so the people in Zoom can hear what you're saying.
All right. Great seminar. So lots of questions, but maybe one to sort of start with, think about, what do you think about the sort of net effect of coral eating fish, right? So like they're consuming some coral, they're spreading symbionts. What's the sort of relative effect of that at population level? And are there other sorts, environmental sources of symbionts besides just the feces of the fish?
Those are really great questions. I think that I don't want to, I'm sorry to give a wishy-washy kind of answer, but I think it probably.
That's what we do.
That's
it's okay.
what we do. I have to lock in with an immediate wishy-washy answer, which is it probably depends a lot on which fish species we're talking about, you know, what the reef looks like already, whether there has been a heat wave or not. Some of these fish, so this is the moment where we're seeing, in my view, we're seeing a general pattern that looks really interesting. And there's different layers of data that I could show you, but now is the moment, I think, to dig in, to try to say, okay, well, what about this fish species and its behavior? What about that fish species? What about this condition? Because in some moments, they might be really helpful, and in other moments, they might just be killing or just like eating coral and, you know, not helping as much. Some fish are really like stick to a very small territory and only go to the bathroom in one place. Other fish have much bigger areas around the reef in which they move, and they go to the bathroom all over the place. So two different species like that. Some fish, when they have a mated pair together, they behave differently than if it's one fish swimming around by itself. So there's a lot of nuance here that I think that we can dig into to try to understand what might be helpful and what might not. Folks who are doing coral restoration, interestingly, sometimes when you're growing baby corals and you want to put them out on the reef, that can be one situation where sometimes these coral consumer fish might not be very helpful to the effort, because in Florida and some other places, when you put, it might be that you're putting something new out on the reef, the parrotfish tend to come and just go, and eat the whole thing when it's tiny. So that's not very helpful to that particular coral that you've put out on the reef. It might spread some microbes. So those are all kind of different nuances. And then I think that, you know, we've looked at a number of fish, but we need to look at reefs around the world and look at more species. And then it's not just fish that bite corals. There are also fireworms, there are crabs, there are all kinds of things that are interacting with corals this way. So I think it's I could keep going, but I'll stop.
I should have pointed out this is a community lecture, so while techie, profound, sciencey questions are cool, any questions are cool. Like, what
Are you
percentage of
saying I
coral
was
reefs are
profound?
what percentage of coral reefs are bleached?
Yeah. But
That's the
what?
question. What percentage of coral reefs are bleached?
Ooh, I don't know a percentage right now, but we are in the 4th global coral bleaching event that has started. And a global coral bleaching event means that there are many reefs around the world that are all bleaching at the same time. So I don't know the percentage right now. Do you know, Anya? But it's high right now. Yeah.
Okay, thanks. You have a question in front.
Okay, thank you so much. It's so educational. It'll take me days to absorb it. I'm curious. Seems like, well, anyway, the summation in my head was, oh, we came to fish emulsion. And I'm teasing a little bit. But that's what I use in my plan. But this is way off into another area, but is anyone anywhere studying the impact of geoengineering on the ocean? Meaning, dropping nanoparticles by the ton into the ocean, near or far?
I don't know. That's a good question. I don't know as much in terms of like nanoparticles specifically, but I would say certainly in terms of different things that we can do at a large scale to the ocean to try to deal with effects of climate change and other things. There are a lot of different things that are being looked into. Certainly not, only fish species and the power of fish, but So there are some geoengineering things like solar umbrellas or seeding the ocean with iron to try to make blooms happen and have, carbon sink down to the bottom of the ocean and draw carbon out that way. So there are a lot of things happening.
Yep. We have a question at the back, and then I'll take one from Zoom. Right, Anya.
I don't know. The answer to that question is no.
There you go.
This is super cool, and I was really interested. It seems like there's a slight bad word, but there's a sweet spot where the fish are still able to kind of exist on these like environments where this leaching currently occurring. But is there like a community tolerance, like thermal tolerance, where at which the fish won't be able to exist on these environments? So it's really this mechanism, like, what's the tolerance the fish can exist at to continue to reseed the coral?
That's a great question, right? Thank you for that question. One thing I will just generally say is, I'm talking with you about a possible solution and thinking about things we can learn about systems and then try to leverage to try to improve things. But this is not like, oh, we just don't need to worry about anything now because fish will go to the bathroom around the reef, right? So exactly to your point, there are, as temperatures warm, and there will be some temperature where fish can't survive biologically, we're unlikely to hit that temperature before we hit the point where there's so much bleaching on the reef that, many, many corals have died, and then fish might not be able to continue surviving. Certain kinds of fish may not be able to persist very long after all the corals are gone. And then you can layer onto that if there have been historical overfishing or current overfishing practices. So there are a lot of ways where you could lose the fish that are going to do some of these jobs. And that's where, maybe considering coral eating fish husbandry, where then you put some of these things back on the reef. Or that's why I even mentioned a robot, right? I would rather have a fish go out on the reef and do the thing that it normally does than put a robot out. But maybe there are certain circumstances where you just put a bunch of corals out and there just are no fish. And so you need, for a time being, it might be useful to have something like a robot mimicking that fish behavior. Yeah.
Adrian, I have a question here from Zoom. Is there any role for genetic engineering or selective breeding of beneficial species?
Well, people are interested in how can we find ways to kind of encourage things to have sex, so to selectively breed them to increase the standing stock of mutations in the DNA of populations of a species. Because if we have more variation within populations of a species, then some of those variants might be more thermally tolerant, right? And then that could get incorporated into something like a restoration effort. So people have talked about doing that, and people have considered out to, first, if we can make some of these organisms into genetically tractable systems where we could consider trying to, genetically modify them, then that's something that could be considered. But just like there are ethical considerations of releasing microbe probiotics onto a reef or robots or other things, certainly there would be a lot of ethical considerations to genetically modifying Symbiodiniaceae or corals and then releasing them in the wild. But the first thing is to probably learn if we can do it, how we can do it in a lab setting, and that we have not even gotten there.
Don't throw that thing at me. So you talked about overfishing. your studies in Moorea, we were just there last year, and the corals are amazing, and the fish are everywhere. Are you seeing in that study site in Moorea overfishing?
I think, some of when you're talking about overfishing, et cetera, there's a historical perspective of how many fish were there decades ago or 100 years ago versus in the last 5 years, et cetera. If you go across the islands, the Society Islands in French Polynesia, you're going to see islands where there is more human influence on the reefs and islands where there's less human influence on the reef. So I think in islands that have experienced, have human populations on them and have experienced more human use and more fishing, there are definitely fish stocks are depressed relative to a more pristine, less used reef. So, it also depends on which fish maybe you're talking about.
I have another question. Oh, and there's one over here. The corals growing in the Arabian Gulf or the Persian Gulf, just either way, are in warm water, right?
Right.
Do we know, is that the coral or is that the symbionts that are so tolerant of the warm water?
So it could be both.
Right.
Yeah. And so I think that if you are comparing populations in a warm area in a Gulf to a less warm Gulf, you will see some population level differences in the coral animal itself. And then you would probably see local adaptation in one population versus another of the Symbiodiniaceae as well. So there can be, even within a Gulf like that, there can be some species, whether it's coral or Symbiodiniaceae, that are extra good at dealing with high temperature. So,
Yeah.
but yeah, overall, in the hot Gulfs, we see adapted thermal tolerant species.
Like I need to throw this.
Ready? Lighter stuff, not so, but when we're monitoring the full coverage or how's that?
Yes. Yeah, that's actually, it's actually hard to follow, well, at least it was hard for us at times to follow fish around for long enough to see them go to the bathroom. It's easier to follow them long enough to see them eat. And then sometimes you see them going to the bathroom. So we would basically try to follow them for as long as we could, at least 5 minutes, but staying far enough back so that we weren't affecting their behavior, and then record everything and just do it enough. We just were seeing it as we were recording it. And then we did some, just as a teaser, we also did some experiments that I didn't show you where, we had those observations of feces falling on corals. We also tried some experiments where we took either feces from the coral eaters or feces from the algae eaters, and we broke, got pieces of the same coral and put the different kinds of feces on one piece or the other and saw what happened. And in less than 24 hours, the algae eating fish feces caused lesions, like it made the tissue die. I'm not going to show you a bunch of stuff, but just because it's so exciting. Here we go. Okay, so just as a tiny example, here's one piece of a coral, and then we put feces from a coral-eating fish on there, and then after 24 hours, we took it off, and in that square, you can see like the same piece over time, and you can see the tissue still intact. And when we did that with another piece from the same coral colony, and we put the algae-eating fish feces on there and took it off, that white is absence of tissue. So we saw some really intriguing patterns, and then we did some other stuff that showed us when we killed the microbes in the different kinds of feces and put them on, it didn't cause that same number of lesions like we saw in the fresh. So we really do think that some of those bacterial pathogens of corals are having an active effect on corals. So it matters what kind of fish is defecating and what falls on a different coral, we think.
Hi, thank you so much for your talk.
I was wondering about how much symbionts vary in one species of coral and geographically. See a lot of variation in symbionts. And then if you do, I thought it's what you're already doing the scientific scale map where you can look at whether symbionts fish are tracking with coral that they're over.
That's such a fun question. And that gets into also, Jay, what you were asking at the beginning in terms of sort of like this different coral species are more or less wedded to having a particular kind of Symbiodiniaceae or maybe a couple of kinds. They don't just have like willy-nilly any kind of Symbiodiniaceae in any coral. And so those are patterns that we think are really interesting. So at a reef scale, sometimes even like inside the reef crest versus outside the reef where there's more activity, sometimes you can see corals of the same species hosting different kinds of Symbiodiniaceae. Some of these fish that we're talking about do or don't exist on both sides. There's all kinds of interesting things to think about in terms of population level variation, et cetera. Yeah.
One more question here on Zoom, maybe I can ask you is, are corals spreading into traditionally cooler waters? And if so, do these fish follow them?
So they're, in general, that would be a tantalizing thing or a optimistic thing if corals could basically just range shift a little bit at least. So if they could go either north or south into waters that are still cooler as climate changes. There may be some examples of this happening at a small scale, but it's not happening at speeds or to the extent that would be necessary for that to be a solution for reefs. And in particular, there have been some modeling studies, so like some prediction studies for which parts of the world could this maybe even happen. And that partially depends on the continental shelf because corals need, if the continental shelf drops off really quickly, then there's nowhere for a coral to settle. There's nowhere for it to move and then settle and still be close enough to the surface to get light and all that. And so, unfortunately, the places in the world where maybe there's even some of that continental shelf that theoretically could let that happen, those are also the places that have the least diversity of corals, et cetera. So that is a great question, but it's not really happening, and I don't know I assume the fish would follow if the corals were doing it significantly, but I think it's not going to be an option, a significant option.
Right. Well, we're going to thank you, but I just want to reiterate, we continue the conversation more informally outside, and everybody in this room is welcome, invited to join us, have some refreshment, and continue talking. And Dr. Correa, thank you again for a wonderful talk.
Thank you. Thanks to you all for coming.